Hi, everybody. My name's Phil Burke. I'm a technical account manager here at LabKey. And today I'm going to be talking to you all about the Biologics LIMS suite that LabKey has. We'll be talking about kind of unifying and connecting your R&D data for making faster decisions throughout the biologics development process. Um, and so today my plan was to spend a little bit of time talking about some of the problems that people typically encounter when they're working with, R&D biologics discovery. Um, and some of the things that we're aiming to solve with LabKey. But for the bulk of this webinar, what I'm actually going to be doing is just jumping into LabKey itself and showing you what's possible with the software. Um, so the first thing I kind of just wanted to talk about was LabKey itself. So I wanted to let you know a little bit more about who we are. We've been around for over 20 years, and we've used that experience that we've had to provide scalable solutions to clients, doing a variety of kind of different types of research. So from academia to research, all the way to biotech and pharmaceuticals. Our support team, so account managers like myself, exist to ensure that the data migrations of customers and onboardings are successful. And we really take a lot of the feedback and guidance that we receive from organizations like yours to basically shape the features that we deliver. And so that is actually why we created the Biologics LIMS product. We were hearing over and over again that there were some real issues that the groups were facing within the scientific community that didn't have, good solutions. So let me just talk a little bit about kind of some of these issues that we were seeing crop up. So the biologics reality is first off, you have very complex sort of entities and relationships, and these can be very difficult to represent with just generic tools. So your data is very unique to yourself, and it's got some eccentricities, and trying to force it into a predefined structure can often be more trouble than it's worth. And then similarly, you might find a solution that works for some of your data or some of your in-lab processes, but not all of it. And so then you end up Frankensteining kind of different solutions together to get sort of an entirety of your research program, accurately represented in whatever software you're using. And this can lead directly to the next problem, which is data being scattered across different sort of locations within your system. So uh, when you finally kind of find the data that you need, it's then difficult to share it with like team members and things like that as well. So you've got data scattered across various points, and then people need to be able to access that data. So another thing that in that sense people run into is that manual handoffs really can increase errors and slow down decisions. So not being able to sort of grant people access who need access to see the experiments and results that you've been doing can definitely cause problems. You want to be able to kind of quickly identify what's viable. And when you can't, this can really just slow things down quite a bit. And then that leads into the final kind of issue, which is that traceability gaps can also sort of cause problems down the line. So when you go back and you audit and you say, what did we do? If you can't really ask questions about your data and see who did what when, then you're not going to be able to get those repeatable results, and you're not going to be able to show that you have a robust process to folks who are interested. So, given all of these sort of challenges, we sought to make the LabKey Biologics LIMS product something that's capable of solving these issues. So LabKey Biologics LIMS. So this is a lab inventory management system or lab information management system. And it's purpose built to basically model biologics entities. So specifically like antibodies, multispecifics, fusion proteins, and just capture those sequence construct, like protein relationships. This is a unified system of record for various entities, but we track your bioregistry, we track your samples, your experiments, the results that you're doing, sort of the analysis on those to an extent. Um, and we keep all of that in one place. And then the other thing that we allow is basically standardization of complex workflows, while being able to kind of configure how your lab runs. So this just involves repeatable templates. Configurable sort of steps and rules and things like that. And then we've got built-in traceability. So you can share various views across teams. You can give role-based permissions access and have like program level dashboards. And then you can also sort of ask questions of your data, like cross-study comparisons, view trends, things like that. And ultimately, the other thing that's built in is just a defensible audit trail. So you can see who did what, when, with what materials across each run and step that you're kind of doing. Um, and so given all of that, I now just wanted to actually jump into a demo of using the software. And so my plan for this demo is I'm going to be focusing on basically modeling biological entities and connecting your data to your samples. So Grab some water first, though. All right. So let's jump over to LabKey. So this is the application itself. And the fundamental point that I kind of want to make in this demo is basically just want to show you how LabKey allows you to build relationships between the molecules that you're studying and your in-lab research processes, and then the data that you're generating. So this is the main LabKey sort of LIMS interface, and you can see there's quite a lot going on here. But this is basically just a landing page that has some high-level experimental details about the things that you're tracking in your system, like the various samples that you have, are the various experiments that you're running. Now to get to the real backbone of the system, I'm actually going to go click up here to the dashboard. And we've opened this up, and you can see that this dashboard is laid out in several parts. The three kind of core parts that I'm going to be focusing on today are the bioregistry, various sample types, so your in-lab samples, and then assays. So basically the experiments that you're running that you're generating data for, and importing that data into this system. Um, so the first things first, I'm going to kind of jump to the bioregistry and just give you an example about that. So let's go to protein sequences. Doink. All right. And so here's just a list of all the protein sequences that we have in our system. And I mean, mine is an example list, so I've only got, 21 or so proteins registered, but you could have as many as you are studying, essentially. And what this is really, as I kind of mentioned, this is a bioregistry. So what we're keeping track of is unique sequences within our system. These are conceptual entities. This is the sequence itself, and the properties therein. This isn't like a sample in a tube. This is the concept of the protein EGFR, for example. And so you can see that in this grid that I'm showing you here, there are relationships between sort of the proteins that we're generating, EGFR. We've got some information about that protein, like its sequence, the length, the folder, sort of project it was generated in, and then some of its biophysical properties. We also have some information about some of the relationships that exist within the system. For example, the nucleotides that could translate this protein. For example, we've got two different sort of codon shuffled sequences that both translate into EGFR here. Um, and you can also see some other properties in this table, but I'm now first going to just jump to an actual protein sequence within the system. So let's go to protein sequence 18 here. Um, so we'll jump over to protein sequence 18. And you can see this is actually a protein component. So this is the light chain of an antibody called olatramab. Yeah. And if we just look at this overview here, we can see a little bit of the information that we already saw on the grid. So we can see again, yeah, this is a signal peptide. In this case, our system automatically recognizes that this is an antibody component. It's a kappa light chain. It's related to a few other things. So for example, it's a component of molecule 1 here. We could learn a bit more about that. It's encoded by nucleotide sequence 18. Um, and so this is, again, just kind of that overview. We could also, take a look at the sequence of this protein. So I'm going to jump over to this tab called sequence. And here we can see the protein sequence within the system. So you can see that this protein sequence is annotated, for example, and you could bring this in from anywhere. So you could basically you could paste this sequence into the system. You could also bring it in by basically uploading a GenBank file and having that be automatically parsed. You could translate it from a nucleotide sequence that exists in the system, and it would automatically build that connection of if you sort of input a novel nucleotide sequence and say, hey, translate this into a protein, it would make a protein and then translate it. Um, and as I kind of mentioned, the annotations that we've added here are also automatically added if you're working with a protein that biologics, recognizes. In this case, this is a pretty straightforward light chain for an antibody. And so all these annotations that exist were manually, or not manually, automatically input for this sequence. And so you could, do other things like copy this. Just going to change the size a little bit to make the whole thing show up. Um, you could edit your annotations, and of course, you could add your own as well. But let's see. So, given that, basically, we've got our protein sequence. Now, I kind of want to jump back and actually talk a little bit more about molecules within the system. These are similar protein sequences. So, I'm going to jump back here. I'm going to jump to this molecule M1. And again, we can see here that, we've got the components of the molecule. So, we see that this is the antibody elatrumab 1. Um, it's got a structure format IgG, so that's recognized. It has some physical properties. We could dig into those more in the physical properties calculator, which I'll just quickly kind of show this. This is calculating a lot of physical properties. You could change what you're doing the calculations on if you modify these ranges and things like that. Um, Getting back to the overview though, and we can also see that this molecule is made up of two components. So protein sequence 18, which I just showed you, and then protein sequence 17, which is the heavy chain of this antibody. And we can see, again, other details about it. So that's just the overview. What's probably actually important is getting the biological information about what this like where this molecule is and how it fits into the rest of our bioregistry and the lab ecosystem. So to get a very high level overview of that, I'm going to hit this lineage tab here. And that'll bring up basically a high level view of all of the things that have been done to this molecule or all of the sort of places that this molecule is involved with. So when we jump to this lineage, we see all these relationships that olaparib has within the LabKey system. We can see it's coming from protein sequence 18 plus 17. And if we want, we can click each of these nodes and get the metadata about that. So again, this is the light chain. We could also click downstream of this node to ES1 and see that this molecule is made through an expression system that's using CHO cells and two constructs that are transfected into those. And then when that expression system has been run, we've actually done two different runs of that, and we can see that it's produced two different batches of protein. So we've got ER1 here, which is a physical sample protein batch, and ER4 over here, that's also a physical sample protein batch. We can actually jump into these further. So I'm going to double click this and pull up the lineage for ER1, this protein sample. And again, we can see things change a little bit. So you can see all of the inputs that went into creating ER1 specifically from this expression system one. So now it actually shows the CHO cells and the two constructs that were transfected into this expression system to create this sample. Um, and so you can get a lot of information and you can kind of quickly navigate the relationships between your bioregistry entities, so the molecule, and the physical samples like this expression system, basically this raw antibody lysate, by just poking around through the lineage. Given this, I'm actually going to jump back to the protein batches themselves. So you can see we've actually kind of moved in the system here. So we started in protein sequences, we moved to molecules, and now we're actually at protein batches here under the sample types heading. And so if I jump back to the overview, we've got some information on this ER1 protein sample. We can see where it's located in the freezer. We can see that it doesn't have any antibodies or aliquots. We can see that it's been used in a notebook. Um, Jumping back to the actual grid, though, I'm going to jump to the protein batches grid where we can see all of the protein batches we've made. So again, here's ER1 in the grid. And we can see that we've also made a few other batches of proteins. The other thing that I was going to do is actually, I'm going to close those two for a second. The other thing that I really wanted to kind of show is that the protein grid itself is totally customizable. So, or really any of the grids in LabKey are fully customizable. So in our protein batches, maybe we're interested in, so the sort of default grid just has the name ER1 and a barcode. If we want to pull in other information about this, for example, like we all know that this came from molecule one, olaratumab, but it doesn't actually show that in the grid. I can bring in that information and I can also remove information that's not that useful. For example, maybe I'm not super interested in sample date or when it was created or who by. So if I jump into the customized grid view, I can remove the sample date column. So I'm just going to remove that. And maybe I'll remove created by. So we'll get rid of those two. And then I'm going to bring in some information about where this sample came from. So if I jump into this ancestor node here, I go to molecule sources, I can just add the sort of molecule that is the parent of this. So I'm going to add molecule there. And then I'll also add that molecule's description. So I'm going to go down and just add description here, which is sort of in this case, I've got the description as the common name of the antibody. So if we update the grid here, now we can see that, yeah, it's ER1. It came from molecule one, which is olaratumab 1 in this case. So just a useful sort of element of the grid. And then we've got this in-lab sample. So this is a physical thing. It exists in the lab. One of the things that you want to do with your physical in-lab samples is use them to generate data, which makes sense. So let's take a look at an assay where we actually sort of test these antibody expression lysates to determine their cytotoxicity on some cells. So I'm going to move back to a different part of the interface now. I'm going to go to plate sets. And so I'm jumping to plate sets, and I've got one that's sort of pre-made. So we're going to go to demo fusion plate here. Okay, so this opens up our plate interface. You can see that I've got sort of four separate plates that all exist. These have already been imported into an assay, so I pre-made these plates, but what we've essentially done, I'm just going to select here, is populated these plates with metadata information for which samples are being tested and their various concentrations by well. So we can see that we're testing the sample ER5, um, ER4, ER3, ER2, and ER1. So basically, I'm just running these samples in specific wells within the assay. And what I can do now, since I've got this populated, is just import some assay data directly. So I'm just going to go ahead and hit import assay data here. We can see that it's offering me an assay called CBPA. If we click back here, we can see that there are several sort of different assays within these tabs. The CBPA assay is basically a cytotoxic assay, just doing a measurement of the effects of these lysates on some cells. In this case, this has been configured to work with plates. So I could actually jump to this assay. Maybe I'll do that. But what I'm going to be doing when I add data for this assay is I'm going to be adding data specifically for this plate set called selected sort of demo fusion plate set. So we'll jump over here. We can see that we've already run several assays. I ran a single sort of assay test demo. So in this case, I'm just going to hit the import data button. And it's going to bring up some information about the assay. We'll call this the demo test assay. It's going to ask me which plate set I want to import information from. In this case, we'll choose demo fusion plate, that one that I just showed you. And then it's going to just say, hey, all right, import your data from a file. And so very quickly, let me just pull up the file that I'm going to import so you can take a look at that. And then we will drag it in and import it. So let's see, not that one. Let's do. Cool. All right, so hopefully you can all see this. This is just basically some example plate reader data that we've gotten. And we can see that there's not really information about what's in what well. It's just direct sort of raw data that we're getting from the plate reader here. But because we've already added the template that has the locations of our samples within the wells in the system, we should be able to just directly upload this data and associate it with each of those samples that we've tested. So going to jump back to LabKey now, and I'm going to drag this demo multiplate file in. So we'll drag that in here. Doink. All right. And I'm just going to hit import, and it should bring that data in. Nice. Cool. All right. So we brought in a bunch of data, which is good. So given that, let's go ahead and see what kind of data that we brought in. So we can see that right now, this is basically, it has the plate. It has the plate set that I tested. It's got these samples that I tested in these wells. And then it has these values, so snap 25 and snap 25 mean and standard deviation. Now, of course, we only had the one value in that plate I showed you, just the raw snap 25 value, essentially. So the system is automatically calculating the mean and standard deviation of these replicates that we're testing here. And let me go ahead. And the other thing we can do, so once we've got data in, there's a lot of ways to kind of sort and parse your data. You can filter it, you can sort it ascending, descending, you can do all sorts of things. So I'm just going to filter for ER1 here, since that's what we're looking at. We could, filter for a specific concentration if we wanted to, but I find it often is easier to just start looking at the charts. So we've got a lot of charting kind of capabilities here. And the first thing I'm going to do is just a standard curve fit chart that we've already built out. So I'm going to click that, and we'll see what this curve fit looks like here. Oh, nice. So this actually is showing two separate assays. So I'm going to very quickly filter my run. This is because I ran the same test earlier, but we just imported demo tests, so we're only going to look at that right now. Cool. Okay, yeah, so we can see that this series basically is just looking at the sample ER1. And actually, sorry, one sec. Okay, so I've actually got this set to series as runs, so it's actually stratifying by different runs. I'm gonna change this to sample ID, so we're only looking at ER1 here and save that chart. And again, so you can see, yeah, for ER1 here, it had, min and max values. It was given some slope. We use a specific curve fit calculation. You could choose a curve fit calculation in your setup. So if I actually jump back here, you can see that I'm using a nonlinear 5-parameter Hill-Richards sort of equation to determine the curve fit. But if we wanted, we could use four parameters, or even things like linear regression or polynomial. Those won't look quite as good, given that this is really data that's very well suited to using these sort of higher parameter curve fits. So I'm going to save that again. The other thing to note is what you're plotting is actually what's going to show up. Basically, what's showing up in the plot here is what exists in the grid of data. So right now I'm filtering for only ER1. If I remove this filter, so I'll actually just allow all of these to show up. We'll see all of the samples surfaced in this same grid. So now we see ER1, 2, 3, 4, and 5 for this assay that we just tested, and all these data are available. The last thing I'm going to do is I'm going to resize this to make it so that it looks a little bit better in an ELN. So let's make it like 400, and then we'll make the width 500. That's too short. Let's do, that's a little nicer. Yeah, that looks good. All right, so let's save that. And I could add a title to this as well. So, we'll call it that. CBP assay curve fits. Save chart. All right, and now I'm going to download this as a PNG. You could also download it as a PDF. In this case, I'll just do a PNG. It's going to think about that for a second. And then I'm going to go ahead and add this to an ELN entry that I actually already have going. So I'm going to open this in a new tab so that we can kind of tab back and forth quickly. But we'll go to a notebooks. And so this is just the notebooks overview page. I'm going to hit antibody profiling here. And sorry. So I've got antibody profiling here. You can see this is kind of the notebook entry that I had all set up already. So I've been working in this, studying molecule 1 and molecule 2, which are linked. If we scroll down, we can see that I've got some results that I'm already putting in here. And I was just going to add these data results that I just generated to this lab notebook. So I'm just going to go ahead and I could just attach this. So I could add this as, for example, if I wanted to drag it in an attachment to the notebook. So we'll just drag in curvefit PNG here. That's going to attach it at the bottom. You can see it's just added this attachment to the file. I could also just put the data directly in. So I'm going to paste that in here. You can see we've added the CVPA assay curve fits to this. And so that's now in my lab notebook entry, and I could update that. And let's actually add a link to this new assay that we've added. So I'm going to add an assay. This was CVPA, and let's add that reference. Cool. Perfect. So now we've actually linked the assay that we just did to this, and I could reopen that assay data here. So this will take me right back to the demo test data that I was just showing you. The last thing that I think is totally worth noting is, so we've got all these data. We have data for each of our lysates, and now we want to see what that data is associated with. So I'm actually going to jump back to our source M1. So this is the original antibody that we were looking at. And if I want to see all the data that's associated with this antibody, I can jump over to this assays tab here. So jump over to assays. And you can see that we've run a bunch of different assays on antibody M1 and in various formats. We've got a couple different protein batches of it. So ER1, which I just showed you, as well as ER4. And we've got data for each of the things that we've run on it. So if I jump to CBPA here, now what this is actually showing you is all of the CBPA assay that we've generated for the ER1 sample over time. So it's not just the run that I did, it's all these other runs as well. And if we again just take a look at the curve fit, I'm going to edit this quickly, and I am actually going to change this to series equals run, which first I'm going to need to add run to my grid. So again, whatever's surfaced in the grid view is what's going to show up here. So we'll just add run, update that grid. We can see that we've run several different things. So let's go back to this assay, and let's just change this to run here. And now we can see that we've got a bunch of results for all sorts of different runs that we've done. And let's just do full width. Nice. So we'll save that chart. And yeah, so this is sort of a longitudinal effect. So each time I've run ER1 in these various assays, so including demo test, which I did today, but also a few other test to demo assays, you can see that we've gotten different results for ER1, and overall, all those results are shown here. And so this is really the roll-up that I wanted to show you, is that as you're doing these experiments, all of the data that you're generating is associated with the molecules that you're studying. So given that, I'm going to jump back to our slides and just kind of close things out with a few other points to chat about. So hopefully, even with kind of that glimpse that I just showed you, you've seen some of the benefits of having this type of all-in-one system. And as just a quick reminder, those benefits are increased accuracy. So being able to track all your data in one place with this sort of full transparency and visibility of where things are coming in. Ease of use. So, it's pretty easy to migrate throughout this software. It's relatively quick to pick up. And it's modular, so you can kind of expand the software with your lab as your needs shift over time. You can always add more types of data, more types of assays, things like that. And ultimately, the third benefit is just the ability to make these data-driven decisions. So you can really quickly gain these insights, like the plotting that I was showing you, through visualizations, but searches, but also just having all that data kind of roll up and be associated with your sample, and finding the information you need when you need it, I think is very important. Um, and so, yeah, with that, just in terms of what's next, if you're interested, if this demo, seemed like something that you wanted to learn more about, just reach out to us. Keep your eye on your email. Reach out. We're happy to schedule a meeting with you. We're happy to kind of work with you to see what your data would look like within the LabKey system. And so, really interested in kind of talking to you going forward and hearing a little bit about what kinds of science you're doing. So. With that, I'll just say thank you again. I really appreciate it and hope to talk to you all soon.